The first thing I want to do is start off with gratitude. I am so thankful to be here today and to give this plenary talk, and I'm thankful for the organizers and the IDSA Foundation funders who have made this possible. I started in this space 6 years ago and have watched amazing progress transpire, and a lot of that progress is because of people that are sitting here in the room today. So I really did want to start with that. Thank you, Amy, for the introduction and for all of the organizational amazingness that you pulled together. So I am Nikki Schultek, and today I'm going to talk about bridging silos. And what does that mean? And essentially, what that is, in my mind, is cross-pollinating different specialties to help accelerate innovation around this topic that we all think is important. Otherwise, we wouldn't be here today. So, as Amy said, I'm the founder of something called Intracell Research Group. I started it 6 years ago, and it is a global research consortium that is aimed at identifying possible infectious drivers of two focus areas. We focus on Alzheimer's disease and asthma. And in 2023, I also had the pleasure of co-founding another collaboration called the Alzheimer's Pathobiome Initiative. This is the first concerted global effort toward really advancing the question. And the question that we're asking is, for which patients could microbial infections be an important driver of their underlying process of neurocognitive decline? And with that, I wanted to share my story. So before I can talk about AlzPI, I need to tell you how I got to be here today. So I want to read a quote to you from my favorite book. And it is that illness is the night side of life, a more onerous citizenship. Everyone who is born holds dual citizenship in the kingdom of the well and the kingdom of the sick. Although we all prefer to use only the good passport, sooner or later each of us is obliged, at least for a spell, to identify ourselves as citizens of that other place. And this is a Susan Sontag quote from a book called The Emperor of All Maladies, which if you haven't had the chance, it's absolutely beautiful, by Siddhartha Mukherjee. And when I read this quote, it really hit home for me, because it's my time in as being a citizen of the other place that has led me to be here today. So in 2015, I had a really tough year. Over the course of 1 year, I developed multiple different autoimmune conditions affecting nearly every part of my body. When people are sick, there aren't a lot of pictures of them taken. They're just not around. But this is one of the few pictures I have from this time. My little boys that are now 12 and 14 were 3 and 5. And I was stricken with a bunch of different things, but these were some of the worst things that were happening to me. My lifelong asthma that had been well controlled on a combo drug that came out, Advair, went to being severe refractory. I was taking up to 80 milligrams of prednisone a day, and I still couldn't breathe. I was awake all night long, night after night, hungry for air. I also developed during this terrible year an incurable bladder pain disorder called interstitial cystitis. It is an idiopathic condition, just like my breathing issue was. And at that time, I also was experiencing terrible joint pain. And the final insult toward the end of that horrible year was that I developed neurologic symptoms that clinically worried my physicians that I could be developing multiple sclerosis. So this sort of part of my life is why I'm here today. And so I'll finish with the terrible story by kind of backing up and telling you the conclusion, which is that I stand here today and I'm well. So I'm getting to use the healthy part of my passport. And it's that story and what the treatment that I endured and the history of my own case that has brought me to the point where I desire to help find out, for some of our most devastating diseases, what is the role of microbial infection? So during this terrible time, I had accumulated 12 specialists. Now, before I became a mom, I worked in pharmaceuticals and biotechnology on the commercial side of things. I was a salesperson. And I had the privilege of working for Pfizer and working in anti-infectives, of all things, and psychiatry. And then I got a job as a clinical specialist after that, doing education on tissue plasminogen activator for stroke in 50 hospitals throughout the state of Maryland, And during that time, I got to learn quite a bit of neuroscience. And I say that that's kind of ironic because when I got sick, I quickly realized that the doctors I accumulated, pulmonologists, urologists, OB/GYN, cardiologists, my goodness, primary care, infectious disease specialists, all of these different specialists I was seeing were very focused on whatever their silo was. So when I'm at the urologist and I'm saying, hey, could my bladder pain have anything to do with this terrible breathing situation that I'm in? And what might that have to do with my joints? The thing that I really noticed that stuck with me was that no one wanted to think more systemically. And the scientists that lived inside of me wondered what had changed. Because prior to this terrible year, I was running half marathons, eating a healthy diet. There was nothing I was doing that should have led me to be as sick as I was. So feeling that my health was deteriorating and I wasn't going to get to watch Owen and Ian grow up if I continued on this trajectory, I started to do my own research. And as I like to say, one Google search effectively changed the rest of my life. I looked up interstitial cystitis, the bladder pain condition, and atypical pneumonia, which was one of the things I was diagnosed with during this terrible year when the breathing situation kicked off. And what I found was a very interesting small study done at Vanderbilt University involving neurologists and a board-certified infectious disease and medical microbiologist named Dr. Charles W. Stratton. And in this study, which was only roughly 18 patients, they tested the urine using PCR looking for chlamydia pneumoniae. Now, chlamydia pneumoniae is an interesting organism. It's the respiratory form of chlamydia. It's an obligate intracellular bacterium. And in this article, while the authors were very clear to say, you know, yes, we found this organism in 80% of these women, it's, you know, far from being able to say that it's causation. However, this organism has the capability of remaining chronic and driving chronic inflammation, and therefore more work should be done. So then I got very curious. I'd had asthma my entire life. As I mentioned, it was well controlled previously on inhaled steroids and long-acting beta agonists. I did a Google search and I came up with a huge and compelling association between this very same bacterium and asthma, particularly severe asthma. So let's get to some optimism after that heavy stuff. So I was given a priceless gift, and that gift is why I'm here today to continue paying it forward. This is Charles W. Stratton. This is the corresponding author on the paper that I mentioned about interstitial cystitis and chlamydia pneumoniae. As it turns out, he preferred to go by Chuck. We did say goodbye to him a little over a year ago. I wish he was sitting here in the audience today because he really would have enjoyed to be here with all of you. But Chuck was board certified in internal medicine, medical microbiology, and infectious diseases. He was very passionate about investigating the relationship between this unusual obligate intracellular chlamydia pneumoniae and multiple sclerosis. And it's his work that I identified first when I was trying to really scour PubMed to figure out what could potentially be underlying my systemic autoimmunity. So I did a crazy thing. I sat down at my computer, totally desperate, anxious, unsure of my future, and I wrote out my whole case for Chuck. And I said, I'm going to email this guy, but he's probably not going to reply. I could see based on his record on PubMed that he was a very well-respected medical microbiologist and ID professional, and you know, I didn't expect to hear back. But he replied within a matter of minutes, actually. And as it turned out, he heard from patients like me on a weekly basis for decades. So what he said was that there was testing for some of the microbes that had been associated with some of the diseases I had been diagnosed with, serology. And he simply suggested that perhaps my primary care doctor or one of my other clinicians could order those tests. So what ended up happening, and as not to make a long story too long for everyone and put us to sleep early in the morning, Chuck ended up becoming my doctor. He was based at Vanderbilt in Tennessee, and I was living in Connecticut at the time. Through blood tests, we were able to determine that I was infected with Lyme disease, Borrelia burgdorferi. I also tested positive for chlamydia pneumoniae, and Dr. Stratton, Chuck, had devised a protocol for treating people like me. And he did treat people that had multiple sclerosis and evidence of chronic bacterial infection with C. pneumoniae, and with a great deal of success over the course of his career, but not without controversy. So I will say that Chuck gave me something that made me very nervous, especially from my time working in the ID space and working on drug-resistant, antibiotics for AMR pathogens, right? Like I launched when I was at Pfizer. The answer that he suggested was treatment with broad-spectrum antibiotics for a very long period of time. A treatment regimen not too dissimilar from the way tuberculous meningitis would be treated. And with a great deal of reluctance, but also feeling completely out of options and knowing that if I continued to feel and be as sick as I was, that I may not get to watch my kids grow up, I took the antibiotics. And what happened was truly remarkable. I ended up having my conditions go into remission. My lifelong asthma went away, and I was able to come off of the inhaled steroids I had been taking for over a decade. And these things that I was diagnosed with, people like me don't usually come back. These stories don't usually end this way. And so when I had this remission over a period of time, I went back to Chuck and I said, what are we doing about this? What if there are other people like me out there that are going down the tubes and they're sick? And the answer for them is treatment, which is able to be repurposed. It's inexpensive. They're treatments that have been utilized for other diseases for very long periods of time. Treatments that someone will put someone on for acne, things like minocycline or doxycycline. People don't currently have access to these treatments that are suffering with some of these conditions. And so this was how Intracell Research Group got started. And we ended up focusing on Alzheimer's disease because it's cruel and because there was a tremendous need and a very strong association with infectious agents that, in my opinion and in the opinion of the people that I work with, like Dr. Brian Galen, one of my board members who's here today, was important enough to investigate. And we also work on asthma because that was one of the diseases that tortured me, and I have a child that's asthmatic. So with that, and earlier what Amari said really stuck, and I want to read you an amazing quote by Einstein, and it's that the formulation of a problem is far more often essential than its solution, which may be merely a matter of mathematical or experimental skill. To raise new questions, new possibilities, to regard old problems from a new angle requires creative imagination and marks real advances in science. And the question that I want to pose is, what causes neurodegeneration? And cases like mine, while case reports can be viewed as sort of the lowest form of science, right, anecdotal evidence, cases do inform us because they provide clues. And we cannot and should not ignore those clues. I'm a living person here today. I don't know where I would be. It has been 8 years since I was treated. I certainly wouldn't be standing in front of this room of all of you if Dr. Chuck Stratton hadn't taken a chance to treat me in a manner which was probably not something that would be condoned by a lot of clinicians. Now with that, I want to be a true scientist and point out that the antibiotics that I took also have very impressive pleiotropic effects on inflammation, and they work on a variety of pathways. So to say that I know that I'm well today because we eradicated certain infectious agents, we can't say that for sure. We can only say that I received treatment that was effective for me. But what it should do for the rest of us here today is have us ask ourselves, okay, what are some of the underlying drivers of inflammation in patients? And something really important to point out, and it's incredibly basic, is that infection, in terms of the human body, is the primary driver of inflammation, followed by traumatic insults and other exposures to things like chemicals. But infection is number one on the list in a very broad sense. Infections can and may fuel the chronic inflammation observed in AD. And we know from work that's been done by some of the members in our consortium, Dr. Rudy Tansey and the late Robert Moyer and William Eimer and others, that amyloid beta has demonstrated antimicrobial properties. And we feel these things are compelling pieces of evidence. Beyond that, and some of you sitting in the room today have done some of this work, there is a growing body of evidence supporting this hypothesis in general, at least supporting that we investigate it with a great deal of enthusiasm. So animal models have demonstrated that AD-like pathology can be instigated by inoculation with certain infectious microbes. Microbes can chronically infect brain and have been demonstrated in post-mortem tissues disproportionately relative to healthy age-matched controls repeatedly. Viral and bacterial illnesses, in terms of large biobank studies, have been associated with the development of Alzheimer's. And then 2 very large nationwide cohort-based studies conducted in Taiwan demonstrated that there was an association with bacterial and viral infections, and that treatment for those infections helped to mitigate risk. And it's something that we're all living through, right, in the last few years. The COVID-19 pandemic is demonstrating that yet another organism, a new entrant onto our landscape of infectious diseases, can and indeed impact neurocognitive change in certain patients. But it's not the same for everyone. And that should be a very compelling piece of evidence. And then finally, I'll get into it in greater depth later, this clinical entity known as reversible dementias should really capture our attention. These are dementias for which infectious agents are identified, and when they're treated, the patient may actually experience a full reversal. So, what has been done in this space in terms of treatment? This is a box from our recent publication that came out in Alzheimer's and Dementia in June. I was very honored to be co-lead author along with Dr. Richard Lathe. And one of the things we wanted to document was what has been done in terms of antimicrobials in Alzheimer's disease. And I've got ID people here sitting in the audience, which I really appreciate. What I want to talk about is precision. This is something that really hasn't been leveraged yet in this space. Because while there have been certain trials looking at antibiotics, looking at antiviral treatments, these trials weren't conducted in a manner that is consistent with infectious disease trials. So, and what do I mean by that? So if you have a patient that has a suspicion for, let's say, meningitis, we need to really first figure out what infectious process is transpiring in that individual. You can't just assume, oh, it's a bacterial meningitis, right? You have to actually figure out what's on board before you can treat it. Bacteria don't respond to antivirals. Viruses don't respond to antibiotics. Antifungal drugs don't work for bacteria. So this is something that really is lacking in the space. There hasn't been a lot of precise work that's taken place, with the exception of 2 entities, Core 388 and Core 588, the drug that had a failure of its primary endpoint, Ginstat, which was coming to market from a company called Cortexime, now known as Lighthouse Therapeutics. They were targeting Porphyromonas gingivalis, the oral bacterium, and gingipains, the downstream inflammatory effect of that infection. However, one thing they did not do with their trials is see what other infectious agents were on board in their patient populations. And ID specialists know this well, but infections can indeed be polymicrobial. So it's something that has really limited the space. Another thing that'll really speak to the ID folks in the room is the considerations on susceptibility of the organism to the treatment, penetration of the treatment to the place that is infected. Crossing the blood-brain barrier is complex. And then route of administration. Antibiotics or antivirals, antifungal medicines may need to be given intravenously, not orally. So there are a lot of things to consider when examining possible progress in the space from a treatment standpoint. So one of the things that I'm incredibly passionate about, and I'll say this keeps me awake at night, this is Table 1 from our publication that came out in Alzheimer's and Dementia in June, and it is a comprehensive list, or as comprehensive as all the queries that we did on PubMed, identifying case reports where the primary driver of dementia of all different types was infectious. So what we found were 22 individual case reports and 2 case series. One 45 patient case series looking at HSV and herpes zoster, and another 19 case series looking at Cryptococcus neoformans. What we found was that this entity, or reversible dementia, has a striking presence in the form of case reports. And what does this mean? This essentially means that these patients had an experience with a clinician somewhere along the way that said, we should do a lumbar puncture and test your cerebrospinal fluid, or we should do serology and test your blood. No sophisticated testing like metagenomic next-gen sequencing was used to detect microbial infections in these patients. And what was incredibly striking was repeatedly the authors of these case reports would say that infectious agents should be a part of the differential diagnosis in people with new neurocognitive change. Now, in these cases, there was a diverse array of microbial infections. There were bacterial infections, fungal infections, viral infections, parasitic infections, and polymicrobial infections. And the majority of these cases had reversals, meaning that once a targeted antimicrobial aimed at what was discovered in CSF or blood was given to the patient, that these patients were able to return in some cases to an MMSE of 30. So the question I ask is this: how many are out there? So if people aren't looking for infectious agents or screening patients that have new symptoms and at the first sign of cognitive decline, we have no way of knowing how many people are out there. And that's the question that we pose today, our group poses, is it's not necessarily a microbe hypothesis of Alzheimer's, it's a different question altogether. We know that certain cases of dementia can be caused by things like neurosyphilis, in these cases caused by Cryptococcus, or other cases caused by viral infections or polymicrobial infections. So knowing that they can be caused, we really should be figuring out who those patients are. So what are the challenges that we all face in this room today? One of the things that we identified as a group in this paper that we published, and a large part of the reason we did this paper together, was this lack of consensus. And I'm sure we're all aware, you have conflicting results coming out of different labs. You have one lab finding human herpes virus 6 and another one can't find it. You have inconsistency in the results, and part of the reason is that there's not an accepted way or a consensus approach around identifying microbial infections in postmortem tissues, biosamples from postmortem contexts like CSF or blood, and then we certainly don't have a really reliable way of finding active infections that are impacting the brain and CNS in living patients. And that is a major limitation. Another one is limited funding. So the NIA has funds earmarked for this work, but it's very small. And in terms of the AD budget, it's a tiny, tiny fraction of the total AD budget. Very thankful for these IDSA Foundation resources and grants, but we need more grant opportunities. And part of the issue that surrounds this whole thing is economics, actually. So for the ID people in the room, they know this well, that the antimicrobial drug space is probably the most struggling drug space in the entire industry. There have been more than 10 antibiotic companies or antimicrobial companies to go bankrupt over a course of a decade. And the reason is that antimicrobial drugs do not command a high price. So companies largely have left their antimicrobial portfolios in favor of pursuing things that are frankly more profitable, like biologics that have to be given to a patient every month for the rest of their life, or rare disease drugs that have enhanced pathways through the FDA. So if this were a thriving space, I truly believe we'd be seeing more innovation in and around the idea of microbial infections as a driver for chronicity, in the brain particularly. Now, one of the things that's a challenge is the different silos in terms of even applying for research dollars. The NIH is divided up into different groups, and technically, our ideas here in this room straddle those groups. But that's actually also a benefit, because it means that we could potentially approach different funding agencies like NIAID, NIA, NINDS, and others, because our work is actually something that straddles more than one. So where there's a challenge, there may also be a possible benefit. And then last, and this is a big one, the way that tissues are collected for research destined for Alzheimer's and neuroscience research is not in a manner that is at all sterile. And some of the objections to this field at times can be about contamination of samples. You know, and this is something that as a group, we definitely need to think about, is the possibility of autopsies that are done in a manner that is, granted, the morgues are not, because we've talked with pathologists, they're not very sterile places. It's never going to be as sterile as an OR, but there are certain things that can be put in place to minimize the risk of contamination. And having access to samples for all of us to leverage that have been collected in this manner would be incredibly beneficial. And with that, I want to talk a little bit about this consortium approach that we have with our Alzheimer's Pathobiome initiative and how it helps to overcome some of the challenges I'm talking about. So interdisciplinary collaboration, time and time again, if you read about innovation, is definitely going to accelerate the pace of innovation. When you bring multiple different people to the table with a diverse array of expertise and different opinions, you can avoid mistakes. You can absolutely bring in novel thoughts and ideas and begin asking questions that may not be posed if you just stayed within your silo. Samples can and should be shared. So this is another huge issue for researchers, right? Accessing patient samples. This was somebody's life, particularly in the case of postmortem samples. As a scientific community, we should really do our best to ensure that we interrogate those samples, that we truly learn as much as we can from those samples. So our initiative is all about maximizing those resources and using as many methods as we can to look at the samples. And then another thing that is a huge opportunity is sharing data sets. So sequencing data can be analyzed using different bioinformatic techniques that reveal new learnings. We could be doing this, and we should be. And then finally, generating a form of a consensus for detecting microbes in patient samples, whether those are postmortem samples or biosamples from living patients, absolutely will aid in understanding how these microbes are interacting with us and how they may be interacting with our brain and CNS. So our idea for Alzheimer's Pathobiome Initiative, or AlzPI, isn't that we all, get together and there isn't any individual work going on. That's not what it is. This is about maximizing exposure, maximizing resources. The individual labs that are collaborating with us still have their own research projects ongoing and their own unique hypotheses. The idea of this collaboration is really about raising the volume on these topics and making some of these resources more accessible on a broader level, and then honoring patients by doing the most that we can with the resources that are so precious. So I have to thank my husband, Brendan Schultek, for being willing to creep around with me on a construction site in Wake Forest, North Carolina, where I live, so that I could take a picture with those silos. but he's a really good sport. Thank you, Brendan. You're not here today. Our effort is patient-centered. Where I began is where my heart is, which is as someone who is sick, who didn't have answers. And that's what the true north is for these projects. But the graphic here depicted really shows just some of the cross-pollinating we're doing, right? And in the center of this, we have our little Alzheimer's Pathobiome logo, but the idea is for the silos to be bridged, and for us to be deliberately diverse, right? So that we have people with different types of expertise, smart people, asking questions, disagreeing with one another, hopefully in a civil way. and then moving research forward. So I want to get very specific about what we are doing. So the Alzheimer's Pathobiome Initiative, very simply, can be stated in 3 steps. The first step we've already completed, and it is to publish our research roadmap, which I mentioned earlier, that came out in June in Alzheimer's and Dementia, entitled Establishment of a Consensus Protocol to Explore the Brain Pathobiome in Patients with Mild Cognitive Impairment in Alzheimer's Disease. So in this research roadmap, essentially we lay out gaps in the research, opportunities, and different testing methods that can be leveraged and refined to come up with a consensus detection protocol. Step 2 of our collaboration is multi-laboratory pathogen detection. So essentially having several different academic labs analyzing post-mortem biosamples, comparing them to one another from the same patient. For example, brain, CSF, and blood from the same individual, looking at load and composition of microbes, using several different detection methods, knowing that some of the issues we identify in our roadmap are that certain pathogens may in fact not be very easy to detect depending on the method being used. They're sneaky and they don't want to be seen, some of these microbes. And then establishing this consensus detection approach in step 2 and publishing it so that others can begin using it. Step 3 is all about translation. And it's interesting, I actually have to give a shout out to Dr. George Perry, who's not here today. But he's the one that said to me, when I called him and I said, George, the way that chronic meningitis has been evaluated by Dr. Michael Wilson, a neuro-ID specialist at UCSF, testing patients' cerebrospinal fluid using metagenomic next-gen sequencing, looking for infections, and then treating patients very precisely based on what they find. I said, we could do that with Alzheimer's disease, right? How do you demonstrate that something is more than associative and could be causative? It's those cases of reversible dementia, right? This treatment approach, which could potentially unveil a subset. And George said, well, you can't just go and treat people. The tests are really, producing variable results. What testing approach would you even use? There isn't even consensus about that. So thanks to George, our group I got very excited as a fast-paced, motivated person thinking about translation. We really backed up and said, before we can get into translating anything into diagnostics or therapeutics that help patients, we really need to have this consensus approach to identifying the microbes in the first place. So once we have successfully finished step 2 of our project, our aim is to actually go to prospective pilot studies, where we would use the consensus detection approach on what biosample was found to be the best biosample representative of the microbiome or pathobiome in that patient, and then treat people who have evidence of infection to see if we can in fact identify more reversible dementias like you see in the case reports. And at a glance, this is a detailed view of those 3 steps in our plan, where you can see that our aim is to really refine several steps along the way. And one of the big things that we've identified, and probably Dr. Balin will talk about it later, right, Brian, is that some of the intracellular organisms may be difficult to demonstrate using sequencing because of their nucleic acids being encased. I'll use the example of chlamydia pneumoniae, which has a very complex life cycle. It has an elementary body form, which is relatively metabolically inactive, not completely metabolically inactive, but it reverts to that state in the presence of antimicrobials, in the presence of threats. And then as soon as the threat is gone, it uses that state to proliferate and spread, and then it can become metabolically active again. It's these sort of unusual organisms that we don't want to miss while we're busy characterizing the microbiome. And so finally, operationalizing this effort. We are incredibly grateful to the Sim Einstein Foundation for some seed grants that have been provided to our group to get started doing this work. And we are already doing parts of our step 2 of the process. So brain tissues, homogeneous, and sequencing data from Dr. Richard Lathe, our collaborator at the University of Edinburgh, Scotland, have been shipped to Dr. Brian Balin at PCOM, who's here today. Brain tissues, identical samples that Brian has have also been sent to Dr. David Corey at Baylor, who's looking at fungal issues in Alzheimer's. And samples identical have also been sent to Dr. Garth Ehrlich at Drexel, who's an expert in molecular diagnostics, particularly bacteria. And then sequencing data has gone to Dr. Rudy Tanzi and William Eimer at MGH and Harvard so that they can apply their bioinformatics to Richard's data set. We aim to publish these results in 2024, demonstrating not only that we can come together as a group to write a paper, that we can operationalize this global collaboration. So, as an overview, and this might sound controversial, but I'm going to stand here today as a patient and say that infectious diseases should be a part of the differential diagnosis in patients with new neurocognitive change. I'm even going to be as bold and implicate psychiatric diseases in that too, where there's a great deal of evidence. I want to say that this work that we're doing goes far beyond Alzheimer's and dementia. This consensus detection approach that we are creating can be utilized to interrogate the microbiome in other diseases, also in other parts of the body beyond the brain. So it is our aim, that this is where we begin, but certainly we are a group that is deeply curious about other conditions like multiple sclerosis, like psychiatric diseases, and beyond. We want to say that we're partnering actively, and I want to hear from the people in this room after today. I want for us to stay in touch. This is a tough niche for many of the reasons I mentioned earlier, and us being in touch with each other, collaborating and lifting each other up helps everyone. So we are partnering with academic centers, clinicians, industry, advocacy organizations, and stakeholder groups. And it is our aim, absolutely, that these innovations don't just end up being really cool basic research. We want them to translate. We want it to translate into accessible diagnostics to diagnose infections, and we want it to translate into, eventually, preventatives and therapeutics. And nothing is possible without a team. And I cannot fit our entire team on one slide, so what I wanted to do was just take some of our core teammates and pull out some of their areas of expertise and areas of focus that they're working on. And we have people, with very diverse array of backgrounds. We have Dr. Lavinia Alberi, who has studied the olfactory route of infections impacting neurocognitive change. She's also an expert in salivary diagnostics. We have Dr. Brian Balin, who's here today, who has looked at the role of Chlamydia pneumoniae in Alzheimer's disease and infectious drivers of Alzheimer's for nearly 30 years. We have Dr. David Corey examining fungal infections. Dr. Corey is interesting because he's also studied the role of fungal infections in asthma and issues with the airway. So his interest in other areas of the body brings a great deal of range in terms of his expertise. We have Dr. Ehrlich, who has been an incredible force in molecular diagnostics and been very involved in the Lyme disease space, which is very close to my heart. And then we have Drs. Eimer and Tanzi looking at the evolutionary role of some of the pathology that's seen in Alzheimer's and the antimicrobial protection hypothesis. Dr. Richard Laith, who is our resident sequencing expert, among other things. Richard has been incredibly passionate about examining the role of microbial infections in neurodegeneration for decades. And Dr. Amy Nelson looking at vascular issues and how vascular issues and infectious processes intertwine to lead to changes in the brain. And I really want to thank our full team of collaborators. I wish they could all be here today, but Dr. Ruth Itzaki, who's a pioneer in this space, looking at herpes viruses. I want to thank Dr. David Hahn, Dr. Wilmore Webley, and other members of Intracell Research Group's board that have been here from the very beginning, including Dr. Brian Balin when I started 6 years ago. And I want to say, to all of you today, to please reach out, to please visit our website and connect with me today and beyond, because the work that we are all doing together is synergistic, actually, and we all face very similar challenges, and we can overcome those more effectively together.